First, I will talk about the solubility of chitosan, because chitosan is only soluble normally at low pH. So at pH 5, all types of chitosan are soluble, so nothing is insoluble. But if you take pH up, a typical industrial chitosan will behave like this. So at pH 6.5, 50% soluble, and at pH 7, very little soluble. And that is a problem because, as I mentioned, cell biologists and others would like to work at pH 7.4 and still have a soluble chitosan. We discovered that Fa around 0.4 and up to 0.6 became more soluble. As you see, Fa 0.6, it's soluble at all pHs. This is very, very nice. So you can use this this chitosan and keep it in solution even at high pH. The reason for that I will not discuss today. I can discuss it with some of you afterwards if you like. Another reason why chitosans are of interest in cell biology and in biomedicine is that it's biodegradable. And one enzyme which degrades chitosan is lysozyme. We all produce lysozyme in the body. It's very rich in, I think, in saliva and in tears, but also in the serum. So chitosans will be degraded in the body. And the rate of degradation depends very much on the composition. So this is a measure of the rate. So a chitosan with a low degree of acetylation, typically 0.1, is slowly degraded. But as we increase, we can increase more than 2 log, more than 100 times the degradation. And this is because the chitosans in this region are not so different from cell wall of bacteria, which lysozyme normally acts on. We have been working on gene delivery for about 10 years, together with many other groups. The point being that if you want to transfect genes, you need to compact the DNA into small nanoparticles. And chitosans and polycations in general can do this. Also, cationic lipids can do this. But cationic lipids are often toxic. Therefore, there is a great interest in trying to use chitosan. So this is the idea. If you have a plasmid DNA, the chitosan make what we call a polyplex, which is taken up by endosomes, release, and finally the gene enters the nucleus and become incorporated in the cell, in the DNA. And this is a study of the compaction process using AFM from my colleague Bjørn Stokke. This is circular DNA. This is also DNA, but not circular. And these are nanoparticles after contact with chitosan. You see how much smaller they are compared to these big plasmids. There is a lot of literature on this. We found, for instance, that the ability to form polyplex depended very much on charge ratio and Fa. So that the very high Fa do not tend to bind very well, but the low Fa have high charge and bind DNA, given a certain charge ratio. This is simple STS, gel electrophoresis. We discovered that the chain length had a big impact. And this is actually in vivo test. Luciferase gene expression in mouse lungs. And we discovered that this is UPC, ultra pure chitosan, high molecular weight, has a certain effect, but the short DP25 and DP about 18 had a big effect, and then for even shorter fragments, there was no effect at all. So very different situation. It is a medium-sized chain that are most effective.